DNA replication. DNA replication to make another copy of DNA. To make another copy of DNA. Where we can find the DNA? Where? You can find the DNA inside the nucleus of the cell. Inside the nucleus. A DNA located inside the nucleus of the cell. And each cell needs to make cell division, mitosis or meiosis. So, DNA replication happens to get another copy of DNA because after cell division, we are going to have two daughter cells and each one is going to receive one copy of DNA. That's why the DNA undergoes into DNA replication. DNA replication happens inside the nucleus because the DNA is located inside the nucleus. And it happens during the cell cycle in the S phase of interphase. The S phase of interphase, what's the meaning of S phase? S phase here is DNA synthesis of interphase to make sure each new daughter cell has one copy of DNA after cell division. How does it occur? How does it occur, DNA replication? How does it occur? As we know, DNA is a double helix, twisted or winded. We need an enzyme to untwist the double helix and unwind the DNA and separate the strands of DNA. The DNA replication, it begins when enzyme called helicase enzyme. Helicase enzyme separates or unzips the DNA. Separates or unzips the DNA by breaking which bonds? Which bonds found in the middle or at the center? They break down hydrogen bonds. Hydrogen bonds. Helicases break down the hydrogen bonds between the bases. Helicase enzyme separates or unzips the DNA. Unzips the DNA. The DNA strands by breaking the hydrogen bonds. By breaking the hydrogen bonds between the nitrogenous bases. It is the first step of DNA replication. And then we have here two strands. And this shape is called replication fork. This shape is called replication fork. We have here two strands separated. They are going to be templates or original strands. Templates or original strands. Each strand is going to form another strand. Each strand is going to form another strand. A complementary strand according to our base pairing rule. A with T, C with G. We need here an enzyme as a builder. Enzyme to get the free nucleotides and form the new strands. What is the name of this enzyme? The builder enzyme or the DNA polymerase enzyme. DNA polymerase enzyme. The DNA polymerase enzyme adds free nucleotides to the original strands and you can say template strands. Here, free nucleotides, they added by enzyme DNA polymerase. DNA polymerase adds free nucleotides to the original strands, and so on till they form two copies of DNA.